This is the Science of GMOs. I'm Jessica Eyes, and today we're going to look at the question, what is gene editing? And joining us is Dr. Bill Muir, a biotechnologist here at Purdue University. Welcome, Bill. Thanks for joining us.
Thank you for having me. It's a very exciting topic.
Yeah, it's exciting and feels pretty complicated. So, let's just jump right into it. What is gene editing?
Well, gene editing, actually, it's a complex process, but in concept is very simple. You can think of our genome as an encyclopedia with books in it and paragraphs and sentences. And gene editing is basically you acting as an editor and say, I don't like the way this sentence is written. And you go in there, you can change words, you can change punctuation, you can do nearly anything. And that's what a biotechnologist does with our DNA, just goes in there and rewrites it. We can insert things, remove things, or add things, and makes it work better.
When people talk about gene editing, I hear them mention CRISPR a lot. So what is CRISPR?
Well, CRISPR, also connected with Cas9, is just a method of doing gene editing. It's like we used to edit manuscripts by what was called cut and paste with scissors, and then we moved to using typewriters with the whiteout, and then we moved to word processors. Well, this is just a word processor. And later something greater will come out. Technology moves at light speed. And next year you'll be hearing something other than CRISPR-2 or something rather, a totally different name. It's just a method of gene editing.
Okay, all right, that makes sense. And is gene editing happening right now in the United States?
Oh, gene editing is happening everywhere in the world. I mean, it's such an easy, although it sounds complex, it's actually quite easy to do, and that's actually one of the concerns is that it is so easy to do. You can do it almost for a science fair project. So it's being used by companies, it's being used by universities. It's very ubiquitous in where it's going on.
Okay, so what genes do we edit? What might be an example of that?
Well, depends upon the purpose. We can literally edit any gene that we want. So now you go in and you ask, just like any business, I need a new product, or I want to solve a problem. And you say, well, how can I solve that problem? And there's problems, several problems in genetics that we had for many years. One example, for instance, is horns in cattle. Horns are pose risk. There's dangers for bulls with horns. Even cows have horns. They'll gore you. They cause injuries and deaths for handlers and also for fellow cows. They fight. Mm-hmm. And so horns have to be removed by dehorning, which is painful and nobody really wants to do it, and it's costly. So the scientists, found the gene that causes horns in cattle. There's a natural gene for what's called POLD. It's just not in all species. So Holstein, which is a dairy cow, doesn't have the POLD gene, so it's hornless. Well, they found basically how to edit the gene to make it hornless, and this was done last year. And now we can have Holstein born without horns and totally avoid that issue. And as a result, the animal well-being and safety of handling animals is greatly improved, and the animals don't have to suffer. That's just one example. There's many.
Okay, thanks for sharing that. And earlier you mentioned that gene editing is actually pretty easy to do, and how that could be concerning. So what kind of oversight is there involved?
There's a lot of regulatory oversight over all biotechnology, particularly in the United States, where we're looking at the process more than the product. So any product, any gene editing or any other way of making a genetically modified organism is if it's an animal, is under the purview of the FDA, the Federal Drug Agency. So even before you start making a transgenic organism, if it's an animal, you have to apply to the FDA to do this research. And if it's a food animal, you definitely have to worry about the containment, make sure it's not eaten and everything until the FDA passes approval that it's not a risk to our health and also that it's not a risk to our environment.
Okay, so you mentioned transgenic. What does transgenic mean, actually?
Well, literally the word transgene means that we're moving genes between species. So a transgenic organism is when we're doing word editing or editing of the genome, is literally moving genes from one species to another. And because we might find a gene that's in one species that was really useful in another, and it just doesn't exist there yet, so we can do that. Gene editing, however, can also be used for editing within a species. So you can have intragenic editing, and people tend to call those transgenic, but they're not really transgenic. They're just editing within the genome. So the technology can be used for definitely a wide range of application.
And this may be a very simple question, but help me to understand this. What is the difference between a GMO, so a genetically modified organism, and then gene editing?
Well,
are they interchangeable?
No, they're not interchangeable. Okay. So a genetically modified organism basically means any organism that's had its DNA modified in any way. It's not breeding per se, because we've been breeding animals forever. Evolution works on genetic variation, so you can call that genetically modified organisms by evolution. So classical animal breeding has been occurring since the beginning of time. But specifically, if we go in and directly modify the DNA of any organism, then it's considered a genetically modified organism, or we prefer to call it a genetically engineered organism.
Okay. And so gene editing is just one of the ways that you can get a
Yes.
genetically modified organism?
Yes.
Okay.
Gene editing is a very specific way. Prior to gene editing, we used things like microinjection, whereby we took what's called a gene construct, a word, so to speak, and we just injected it into the embryo, the day-old embryo, and hoped and prayed that somehow this gene got inserted someplace in the genome and that it would express. And so this is kind of random insertion. You can imagine it had a lot of failures, only about 1 out of 1,000 worked, and we never knew where the transgene was going to be inserted. So gene editing gives us the way now of saying exactly where we want this gene inserted. So it's a lot more exact and precise way of doing genetic engineering.
So I've heard that gene editing is used not just in seeds and food, but also in animals, but also for health. Is that true?
We can use gene editing for any purpose. There are genes that you have birth defects. There's a number of known diseases, Huntington's, chorea, and several of these genes that we inherit that cause a lot of suffering. And technically, we could apply these same technologies to humans for addressing health issues, and this is actually one of the things being looked at. But now we enter into the realm of ethics. Is it ethical to modify genes of humans? We've definitely done this in pigs, for example, where pigs were very susceptible to PRRS, porcine respiratory disease. And there was no vaccine for it, and there's no way of getting rid of it other than once your herd got PRRS, you had to euthanize all the animals, disinfect the building, and start over.
Wow.
Yeah. So now we can, with gene editing, we found out how to remove the receptor for the virus. So the virus causes this disease, but most viruses require what's called a receptor, a place to land, a place to attach to. And so they knew what the landing place looked like, and they simply removed the landing place. And without any place to park the virus, it simply says, I can't infect this individual. So now we have genetically modified pigs that don't get PRRS disease. So this is just one example. Mm-hmm. And this is actually might be one of the first examples of where gene editing is going to move into production phase, because there's already a pig breeding company that has this in the wings and is seeking regulatory approval to produce animals that are resistant to PRRS.
Oh, great. That's really interesting. And so in regards to humans, though, the debate is ongoing.
Oh, this is a huge debate. Anytime we talk about editing the genome, editing genes of humans, then we get into eugenics and breeding superhumans, and it's a slippery slope. Some people, ethicists, will tell us, well, if it's okay to modify genes for disease, how about genes for height or appearance or eye color, or how about IQ? Where does the whole idea of gene editing stop? Can we make a very hard line? This is the only things that we're allowed to do. But once we start that, can we really stop doing this? And so the whole issue of gene editing in humans is very hotly debated. On top of that, in order to produce gene-edited humans, we have to manipulate the embryos. The in vitro fertilization, we know how to do that, but now we have embryos that are like in banks, and several thousand of those will probably be used for test cases to find out how to do gene editing in human, and almost all of those will be destroyed. So this is the ethical issue now. You're going to fertilize these embryos and you're going to inject the gene, and a few days later you're going to destroy them. And this raises a lot of ethical concerns to a lot of people.
This is hypothetical, though. It hasn't happened?
No, it actually has happened,
Okay.
but not in the United States.
Not here?
Not in the United
Okay.
States yet. The NIH has came out and said that it will not fund any research related to human gene editing. But other places in the world have already tried this and are trying to do gene editing in human to address some genetic diseases. And so there's a lot of controversy over that.
Okay, great. Bill, thank you so much for joining us to help unpack this complicated topic. Again, this is the Science of GMOs, and today we've been looking at gene editing. Thank you.
Thank you.